Okay, for part 2 of enzymes, we're going to look at different factors that affect enzymes. Enzyme concentration, the substrate concentration, the temperature, pH, and different inhibitors. Taking a look at temperature, there is an ultimate temperature at which the enzymes work for humans, and that's at 37 degrees Celsius, which is approximately average body temperature. At lower temperatures, the reaction of the activity is typically slow for the enzyme, and that's because there's a low amount of kinetic energy. The substrate doesn't collide as often. As the temperature is increasing, the kinetic energy is increasing, and so the enzymes and substrate molecules move faster. This allows for more collisions. When enzyme and substrate collide with more energy, it's allowing for bonds to form and break. Then it reaches a point where the temperature starts to continue rising, but then the reaction is going to decrease. And this is because now you're increasing that kinetic energy, but at a higher temperature, the enzyme cannot function. It starts to change the shape, alters the shape of the active site, making it weaker. And what's happening is your enzyme is beginning to denature. And so it reaches a point at a temperature that's too high that it is completely denatured. In looking at how temperature affects the rate of reactions with enzymes, there's something known as a temperature coefficient. And a temperature coefficient is represented by Q sub 10, which just means that for every 10 degree in Celsius increase, the rate of the reaction usually doubles or triples. However, there is a maximum temperature before an enzyme will begin to denature. The optimal temperature for an enzyme-controlled reaction will be a balance between the Q10 and the denaturization. This is represented by a ratio. That ratio is going to be the rate of the reaction for your temperature plus the increase, the 10-degree increase, divided by the rate of the reaction of the starting temperature. So you have that starting temperature and then the temperature that you started at plus 10 degrees. Usually this ratio is going to be 2, which means that every time the temperature increases by 10 degrees, the rate of the reaction is going to double. Different enzymes function in different organisms in different environments. Again, when we look at humans, human enzymes like to work between 37-40 degrees Celsius. There are some archaeobacteria that you'll find in thermal vents. They like to work between 70 degrees Celsius. Some can even go as high as 100 degrees Celsius. There's a cold water fish that survives at 30 degrees Celsius, but if it gets in water temperatures higher than 30 degrees Celsius, the enzymes would begin to denature. Taking a look at the effects of temperature, the optimal temperature, again, varies for different organisms. For humans, it's between 37 and 40 degrees Celsius because, again, the average body temperature is 37 degrees Celsius. If the temperature goes higher than this, you're going to have increased levels of molecules where the bonds are breaking due to the increase in energy. The temperature's going up. This is going to cause the hydrogen and ionic bonds to become weak and break. As the temperature continues to go, the enzymes are going to begin to denature. They're going to lose that 3D shape, which is something you find in the tertiary structures. You're going to alter this tertiary structure. If temperatures are colder, the molecules are going to move at a slower rate. There's not as many collisions that are occurring between the enzyme and the substrate. A lot of biological laundry detergents contain enzymes such as lipase and protease. Enzymes that are going to also be found within the body, but they use these enzymes as a cleaning agent. Lipases will help break down fats and oils, while the proteases are going to break down protein chains. Looking at changes in pH, pH is a measure of the concentration of hydrogen ions in solution. Adding or removing hydrogen ions will change the pH. Lower pH has more hydrogen ions. Higher pH values have fewer hydrogen ions. There are bonds between the terminal amino group, the carboxylic group, and between the various R groups. If the pH changes, then the ionic charge on the acidic or basic groups will change. This is going to affect either the secondary, this represents secondary, or the tertiary structures. And then this can denature the proteins. Buffer solutions, which are used to maintain a pH. They have specific pH values that they work at. Buffer solutions can be used when you want to test the effect of the pH on the rate of the activity. You can make buffer solutions at different pH ranges in order to see how a particular enzyme is going to work when it's in a different pH environment. For the optimal pH, most human enzymes work at a pH range between 6 and 8. Depends on the localized conditions. For examples, the stomach has pepsin, which works at a pH in a 2 to 3 range. Trypsin, which is found in the small intestines, works at a pH of 8. And then the salivary amylase, which is found in saliva, works at a pH of 7. Taking a look at a graph, these are 3 enzymes that you should be familiar with. Pepsin, which is found in the stomach, likes a pH closer to 3, but anywhere between that 2 to 3 pH range. Salivary amylase, which is found in saliva, works best at a pH of 7. And then trypsin, which is found in the small intestines, likes a pH between 8 and 9. Foods also contain enzymes which start the breakdown of the food. If you want to slow this process down, then you could add vinegar or lemon juice to the food item. That's going to lower the pH value and as a result slow down the reaction so the food doesn't decay as quickly. All right, some questions for you to answer. If you would please make sure that you answer these before continuing with your notes. Let's take a look now at enzyme concentration. What's happening when the enzyme concentration is increasing and the rate of the reaction is increasing. This is a reaction that's going to begin quickly, which you notice with your steep curve that's occurring, and then gradually it's going to slow down. The curve is going to level off. And that's because even though the enzyme concentration is increasing, there's only so many substrates that can actually bind with those enzymes. This is showing you as you have an increase in the enzyme, you're increasing the rate of the reaction. More enzymes, you're going to have more collisions with the substrates, but the reaction again is going to eventually level off, and that's because the substrates become the limiting factor. Once all of your substrates are used up, you still could have enzymes there, but if they can't find a substrate, you're not going to have the reaction continuing, and that's why it's going to eventually level off. This graph is showing an investigation using the enzyme catalase and its substrate, hydrogen peroxide. The catalase is present in the extract made from celery. Different concentrations of the catalase solution were added to hydrogen peroxide solutions. The quantity of the hydrogen peroxide, the substrate, used was the same at the start of each one of the reactions. The second graph is showing you the rate of the reaction in the first 30 seconds was calculated for each enzyme concentration. Now looking at how the substrate concentration increasing is going to affect the rate of the reaction. Again, you have this curve that's occurring where you have the increase in the curve, it's occurring very quickly, and then again it's going to eventually level off. This is because as the substrates increase, there's an increase in the rate of the reaction. More substrates mean more frequent collisions with the enzyme. Keeping the enzyme concentration constant, there's going to come a point where every enzyme active site is full. When this occurs, the rate of the reaction will actually level off. This will be the maximum rate of reaction. Each of the enzymes have active site engaged. The enzyme is said to be saturated. Adding more substrate, the enzyme cannot work faster. The enzyme is working at a maximum rate. This is what's known as Vmax. V is representing velocity or speed. It's the theoretical maximum rate of an enzyme-controlled reaction when all the active sites of the enzymes have been occupied. All right, question time, if you would answer this question before moving on. Comparing enzyme affinities. Affinity is the measure of the strength of attraction between two things. For enzymes, it is the attraction between the enzyme and the substrate. The greater the affinity for an enzyme to the substrate, the faster it works. If the affinity is low, the substrate may leave the active site before a reaction takes place. A typical enzyme molecule can convert around 1,000 substrate molecules per second. Carbonic anhydrase is one of the fastest. It can remove 600,000 molecules of carbon dioxide from tissue per second. This is about 10 million times faster than the reaction could occur without the enzyme. A buildup of carbon dioxide could be lethal very quickly. Okay, another question, if you would take a moment to answer this one. Let's take a look at the Michaelis-Menten constant. Michaelis-Menten, which is abbreviated capital K subscript M, the Michaelis-Menten constant is the substrate concentration at which an enzyme works at half its maximum rate. So we have half Vmax. Remember, V was the maximum, so we're looking at half the maximum rate that is used to measure the efficiency of an enzyme. The lower the values of the Michaelis-Menten constant, the more efficient the enzyme. Okay, here's a graph representation for the Michaelis-Menten constant or Michaelis-Menten rate law. Again, this is the least amount of substrate needed to make the reaction go at the maximum rate. The smaller the Michaelis-Menten constant, the greater the affinity the enzyme has for the substrate. Remember, at Vmax, this is when you had your enzyme and substrates were bound together. That's when it was saturated. When you're looking at the Michaelis-Menten constant, it's going to be half that amount. We know that at Vmax, that was the maximum rate of the reaction possible. This is representing your Vmax. That's when the reaction is going to plateau. Your substrate and enzymes are matched, bonded together at a saturation point. At V half max, okay, where you take half of that amount, this is your substrate concentration at which the enzyme has the greatest affinity for the substrate. This is what's going to represent your Michaelis-Menten constant or Michaelis-Menten rate law. The smaller the Michaelis-Menten constant, the greater the affinity the enzyme has for the substrate. So you're going to get a lot of reactions occurring. All right, a few more questions if you would answer these before we move on. Let's talk about enzyme inhibitors. There's competitive reversible inhibition. It is possible that a molecule with a similar shape to the substrate can enter into the enzyme's active site. This would inhibit the enzyme from doing its function. If an inhibitor molecule binds even briefly to the active site, then when it comes out, there's a competition between the inhibitor and the substrate for the active site. If enough substrate is present over the inhibitor, the substrate molecule will win, if you will, and the enzyme's function is basically unaffected. There's enough substrate to bind with the active site. If substrate molecules decline, or there are more inhibitor molecules than the enzyme's function is going to be inhibited. This is known as a competitive inhibition. It is reversible if enough substrate becomes available. This is an example of a competitive inhibition where, if you look at the enzyme, notice the shape of the active site. This is the competitor inhibitor. Notice its shape. And then notice your substrate shape. They are very similar. Here is a case where we don't want to increase the substrate. We want to increase the competitive inhibitor. As for someone who has ingested ethylene glycol, which is a compound found in antifreeze. Ethylene glycol is rapidly converted in the body to oxalic acid, which causes irreversible kidney damage. The active site of the enzyme, which converts that ethylene glycol to oxalic acid, will also accept ethanol. Ethanol is going to act as our competitive inhibitor, which will slow down the ethylene glycol so that it can be excreted. So we don't want to increase the substrate in this example. We're wanting to increase the competitive inhibitor so that the body can get rid of that ethylene glycol. Competitive reversible inhibitors. The inhibitor and the substrate compete for the active site because they have similar shapes. In the case of penicillin, an individual takes penicillin. The penicillin blocks the enzyme that bacteria use to build their cell wall. The bacteria are not able to survive without the cell wall. Increasing the substrate concentration so it can be reversible. Saturating a solution with substrate so it can outcompete the inhibitor for the active site on an enzyme. Another example is a drug by the name of disulfiram. Disulfiram is given to individuals sometimes that have individuals with a drinking problem. And so to help them overcome their drinking habit, they will take the medication or drug called disulfiram. And what that does is it prevents the alcohol from being broken down, and it produces different compounds that makes the individual very sick, and they begin vomiting to discourage them from drinking. Now let's look at non-competitive reversible inhibition. The inhibitor molecules bind to another part of the enzyme, not the active site. So we have our enzyme, here's our active site, but then I have a molecule that's going to bind on another spot, another opening, and this is what's known as an allosteric site. When another molecule binds to the allosteric site, it affects the normal arrangement of the hydrogen bonds and the hydrophobic interactions holding the enzyme molecule in its three-dimensional shape. As a result, this changes the active site. While the inhibitor molecule is attached to the enzyme's function will not occur. This is known as non-competitive inhibition. There's not any competition because competition occurred when you had an inhibitor trying to bind at the active site along with the substrate. This molecule is binding at the allosteric site, so it's a non-competitive inhibition. Increasing the substrate concentration will have no effect on this inhibition. Inhibition of an enzyme can be harmful or even fatal, but inhibition can be essential. For metabolic reactions, they must be controlled so that no enzyme can be allowed to work without stopping at some point. Otherwise, more and more product would constantly be made. Non-competitive reversible inhibitors, again, inhibitors that are going to bind to another spot on the enzyme other than the active site. They're going to bind to the allosteric site. They're known as allosteric inhibitors. This does cause a change in the active site. An example of this would be anti-cancer drugs that inhibit enzymes that are involved in the DNA synthesis. Looking at the effect of inhibitors on enzymes, your competitive inhibitors slow down reactions but still allow them to occur with a surplus substrate. So our normal enzyme rate of reaction, we have our curve as the reaction's occurring, eventually plateauing. A competitive inhibitor, as long as you've got enough substrate to bind with the enzymes, it slows it down. It's not as fast as the normal enzyme, but it's still able to proceed forward. Non-competitive inhibitors change the shape of the enzymes so they no longer work. Therefore, the rate of this reaction is going to be even much slower. You're not going to have as high as a rate of reaction with your non-competitive inhibitor. Looking at feedback mechanisms, an end product inhibition can be used to control metabolic reactions. The end product inhibits the enzyme at the beginning of the chain of reactions. The enzyme slows down as the amount of end product increases. The end product can lose its attachment to the enzyme, so it gets used somewhere else and the enzyme returns to its active state and makes more end product. So you have a substrate combines with enzyme 1 to make a product. And then this product becomes the new substrate to combine with a new enzyme to make a new product. And then that new product combines with a new enzyme to make an end product in the end. But instead of this reaction continuing to have a chain reaction, this end product, our third product, which is known as our end product, and it could have a longer chain here. This end product can come back and bind with enzyme 1 as an inhibitor. Well, if it binds with enzyme 1 as an inhibitor, then our substrate doesn't have anything to bind with. So as a result, product 1 is going to decrease, which is going to cause the second product to decrease, and therefore the third product to decrease. Once the third product decreases, then it reverts back to its normal state, where the substrate can then bind with enzyme 1. This is what's known as a feedback mechanism. In feedback inhibition, the product form eventually inhibits the reaction from continuing. Once the product becomes too low in the cell, production will begin again. Irreversible inhibition. Inhibitors permanently bind to an enzyme. You can have a competitive inhibition that's irreversible. It permanently binds to an active site. You can have where the allosteric site is permanently binds to an allosteric site. As a result, it's going to permanently change the shape of the enzyme. Things that would be examples of irreversible inhibition would be the nerve gas sarin and many insecticides. Nerve gas is used in warfare where it's going to deactivate the neurotransmitters. It actually binds to the active site and then deactivates that neurotransmitter, which is needed for nerve impulses to be passed from one neuron to another across the synapses. If the enzyme is inhibited and the nerve impulses cannot be stopped, this is going to cause a prolonged muscle contraction. Paralysis can occur, even death could result since the respiratory muscles are affected. With insecticides, they work in a similar way on the insects, which can also lead to harm to the nervous and muscular system of a human if they're overexposed to them. This is just a quick little diagram, again, showing you the enzyme inhibitors. The competitive inhibition, where you have your substrate and an inhibitor that are going to both compete for the active site. If the inhibitor is going to bind to the enzyme, then the substrate is not able to bind with the enzyme, so we will not have a reaction. And non-competitive inhibitors, where you have your substrate again and your inhibitor, but your inhibitor binds to that allosteric site, which is going to alter the shape of the active site and prevent the substrate from binding. Irreversible inhibitors, where you have something that's going to alter, again, it's going to alter that active site, preventing your substrate from joining. And then when we look at our feedback mechanisms, where you have a chain of reactions occurring and the end product is going to come back around and bind with enzyme number 1, which is going to inhibit the pathway. However, once the product decreases, the reaction will return back. Immobilizing enzymes. Enzymes are expensive. They are used in medicine, food technology, and industrial processes. Food companies immobilize enzymes and therefore can use them over and over. Trapping and immobilizing them means enzymes do not end up in the final product. They are more tolerant of temperature and pH changes when immobilized. An example would be the making of lactose-free dairy products. Lactase can be immobilized using alginate beads. The substrate of lactase is disaccharide sugar lactose. Milk runs through a column of lactase containing beads. The lactase hydrolyzes the lactose in the milk to glucose and galactose, making it lactose-free. If milk was mixed with lactase, then the lactase would be gone and the milk would contain enzymes. Using immobilized enzymes, the milk is enzyme-free and you can keep reusing those enzymes. Compounds which help enzymes, they're activators. You have cofactors, which are non-proteins. They're small inorganic compounds. They work with magnesium, potassium, calcium, zinc, iron, and copper. Hemoglobin is aided by iron, and chlorophyll is aided by magnesium. We also have coenzymes. Again, they are non-proteins, but they are organic molecules. They can bind temporarily or permanently to an enzyme near the active site. This would be many of our vitamins, such as niacin, vitamin B3, riboflavin, vitamin B2, or coenzyme A. All right, one more set of questions if you guys would take some time to go through and answer these questions.